Hi, here I am going to give you just a small introduction about myself. Myself Neha Gwal, and presently I am an online teacher and a content developer, and I like to teach the students. It's my huge passion for teaching the students, and students also like the way I teach and the way I plan the lessons for them. My highest qualification is MSc Biotechnology, and I'm also GATE qualified in the subject of biotechnology. As far as experience is concerned, I have very good experience in both the online and the offline teaching, and I have my software already for online teaching with the Wacom pen and tablet. You can have trial session or you can have the trial classes with me. If you like the session, then it's good, and I am very sure about that you will like the way I teach and the way I make you understand the topics which are very tough for you. Even the toughest topics you can be able to understand very easily. Now it's a small introduction about teaching. Teaching for me is just a nurturing of the child. It's not just the completion of the course. With the teaching, you are making a future of the child. So in my way, you are it gives me immense satisfaction when you teach the child and that student is getting everything that you are teaching it. He is getting higher marks and he is getting into the subject, he is developing interest into the subject. It's all depends on the student and the teacher both. It depends on it's a two-way process, depends on both the teacher and the student, that how he grasps what the teacher is saying, and it depends on the teacher how he is getting to the student, and how he or she has to teach the student so that he can grasp as much as he can. Nowadays, the digital learning or we can say e-learning is in progress, and everyone is focusing on online education because it's in boom, and it can be operated from home at a suitable time, and teacher and student both are comfortable with the online teaching. So I'm going to teach the lessons online, and it will be I'm sure that it will be better than any offline coachings where you have to go and you have to take the class. In transporting only, you are wasting so much of time and energy. So at home you can have a much better learning. Now I am just starting off my topic of 12th grade biology. It's uploading. It's a 12th grade biology, and it's the topic which is called genetics and evolution. It's a very important topic from 12th class. You can have this demo and you can find if you are understanding the topics well, so you can go further with me confidently and with full trust. So I'm just giving you the outline of the chapter, and after the completion of the chapter we will be able to know the following things which you have read in 10th and 9th class, also in the lower grades, but those were just the basics of it. I'm going to give you some more description of it and revising the basics of the previous classes also, and then covering the details of the topic of the from the 12th class. So outline of the chapter, it basically covers the basics of heredity, the basics of Mendel, and just the Mendelian traits, the causes and the causes of the Mendel results, and the incomplete and the codominance. What are the reasons for the incomplete and the codominance, why they occur, and then the chromosomal theory of inheritance. And finally, the main which we will learn is about pedigree analysis, how we analyze the family history, the disease which is occurring in the family history. So first of all, we comes to the basics of heredity. So here is the basic of heredity. Heredity, as you know, it's the inheritance of character from the parents to the offsprings. And as we know that each and every nucleus, it has bound DNA within it with the histone proteins, which we have read in the previous classes, and with that nucleus only, in the nucleus only the DNA is present, and in the DNA there are present the genes. So here we can see the diagram is showing, like in the DNA we have a chromosome. In the chromosomes the DNA is present in the coiled form, and in this DNA the various genes are present, which are the basic units for carrying any characters, for defining any characters of an organisms. As we see that genes control the expression of the characteristics. So as we have learned that in the genetics only, the Mendel is the one who introduced us with the factors, which he called the factors, now we call it genes. He introduced with the genes, and these genes are always present in the pair form, one from the mother and one from the father, and these pairs of genes are known as alleles. So all the alleles, one from mother, one from father, are the main basic characters which define the inheritance of the characters from the parents to the offsprings. So inheritance is due to the fact that both father and mother contributes to the equal amount of genetic material to the child. For each trait there are two factors, one from father and one from mother. Then there comes the genotype and the phenotype. What is genotype? Is the whole genetic constituent of any organism, the whole genes, the whole number of genes, the whole matter of genes which are present, and these genes only, they describe the physical characteristics of the organisms, and which we call as phenotypes. So genotype is a set of genes that is found in the individual, and phenotypes is the observable characteristic of that organism. Like genotypes is for tallness, the genes are TT, so that is the genotype, and phenotype we are which we are observing, like tall plant we are observing, that is the phenotype, and the genes which are contributing to it are the TT, the genes characters. So those genes constitute the genotype, and the observable characters constitute the phenotypes. As we have seen in this diagram, this is a mother, this is a father, and this is a child. So the child can inherit characters of any of them, the mother and the father, or the both of them. If the mother is angry in nature and the father is quiet one, then child may be quiet, child may be angry in nature. All depends on the genes, the nature, the physical appearance, that all genetics. Now we come to other basic characters of the Mendel, which we call the Mendel laws of inheritance. We will be reading the details of the laws later in this chapter. First, we will just concentrate on the terms for the introduction of this chapter, that what is genetics, what is inheritance, what is variation. So genetics is that branch of biology which deals with this inheritance and variation. And inheritance, what is that inheritance? That is the characters which are being transferred from the parents to offspring, from one person, from one generation to the next generation. What are variations? Variations is the degree by which these differences are being occurring, the degree by which progeny is differ from its parents. So all this study, they comes in genetics. George Mendel was the scientist who first time conducted the experiments to understand the pattern of inheritance of variations in the living beings. Now we will come to know about the Mendel experiment material, how what all material he collected to perform the experiments. He used garden pea plant, which scientific name is Pisum sativum, for 7 years. For 7 years he conducted the experiments with garden pea plants and proposed the laws of inheritance. And these pea plants, why he selected? Because these pea plants have various degrees of traits, of contrasting traits, and they are very easily available. They can be pure breed, the true breed, and the crossbreed to perform the experiment very easily. And many varieties are available with distinct characteristics. So he chose garden pea plant for his experiments. Now we come about some basics about the Mendel experiments, like I have told you, like why these genetics is important, why the study of this genetics is important, because as we see that the children, they are being similar to the parents. They are they are just by face also, by nature also, they are resembling the parents. So that is all because of the genes where they they are getting from the parents, and this is all the genotype which is being transferred from the parents to the offsprings. And this all gene study is done by George Mendel for the first time. Second basic is every gene which the Mendel take is in the paired form. If we take a green seed, it's the pair of genes, it's a pair of characters of a gene. Like if we take a green gene, then it is capital Y, small y. So why this always the genes occur in pair, the alleles occur in pair, because one from father and one from mother. Mendel first needed to be sure which genes each parent carried. Since pea plants are naturally self-fertilizing, purebred strains were readily available, and purebred plants with green seeds only produce the offspring with green seeds. Then the third basic is then that there is no blending of any traits. Means you will not see that yellow seeds are being blending with the green seeds to giving some other color. There is no blending. That's why we call it pure breed, and that's why we are able to find out the laws of inheritance. Now, the next basic is that when there are two pairs of characters, one is always the dominant one and one is the recessive one. The dominant one is the one which masks the recessive one and which only appears, which is only the observable character for anyone. Like if we cross yellow and green seed, then green is recessive and yellow is dominant. So in the F1 generation only we will be able to see the yellow seeds, no green seeds. How and why, we will be just reading this in the next presentation, that how these experiments are being conducted, what are the reasons for these experiments, and what conclusions we derive from these experiments. So these are all the basics of the heredity which we have to consider about. Now we come to the second presentation. As of now, this is for the basics of heredity in the first presentation. Thank you. So as we have discussed earlier that Mendel take the pea plants and his various traits for the experiments, he basically get 7 traits for performing the experiments, that are stem height, as you can see here, the stem height, the flower color, the flower position, pod shape, pod color, seed shape, and the seed color. For these he has got the contrasting traits like tall and dwarf, violet and white, by which it is easy for the Mendel to perform the experiments with the pea plants. So Mendel experimental procedures, as we see that what is the procedure for the Mendel experiment is he observed one trait or character at a time. The first main thing is one trait at a time. So first he gives the monohybrid cross results, then he done the experiment for the dihybrid cross for the two traits, and then he get the result. So first he took only one trait, that is for getting the monohybrid cross, and after getting the results of the monohybrid cross, he took two traits for dihybrid cross and getting the results for it. So Mendel hybridized the plants with alternate forms of a single trait, the monohybrid cross. The seeds produced by the crosses were grown to develop into the plants for the filial progeny or the F1 generation. He then self-pollinized the tall plants, and then he get the F2 generation. So one trait at a time. In the later experiments, Mendel also crossed pea plants with two contrasting traits, as I was telling you, as a dihybrid cross. And then he self-pollinated the F2 plants also. So this is an image showing the Mendel traits what he has taken, like the shape of the seeds, the round and the wrinkle, the color of the seeds, seed interiors, the yellow and the green, the purple and the white flowers, the inflated and the pinch ripe pods, then the green and the yellow unripe pods, eggshell and terminal flowers, long and short stems. These are all the traits the Mendel has taken for its experiments. Then he did the monohybrid cross with the one trait, and with it he developed the result for the monohybrid cross. What he has done, Mendel, in his first hybrid experiment, took pea plants with tall and the short characteristics and produced a progeny from them. As you have seen, we have taken here the Punnett square. With the Punnett square only it is possible to perform the Mendel experiment, to declare the results of the Mendel experiments very clearly. For if we say that A is for the tall plant and small is for the short plant, in the first filial generation we get only the tall plants, and then in the next filial generation we got the tall and the short plants in the ratio of 3:1 for phenotype and 1:2:1 for the phenotype. Here we are showing one more image which shows the monohybrid cross, the cross of the round and the short seeds. What we got in the first filial generation? Only the round one, because round is the dominant one. Then in the second filial generation, after self-pollinating the first filial generation, we get both the round and the wrinkle seeds in the ratio of 3:1. And here we can see the genotype. This is 1:2:1, as 1 is for S's, the 2 is for S and small s, and for 2 small s this is 1. So genotypes are 1:2:1 and phenotypes are 3:1. Then he performed for dihybrid cross, in which he had taken two traits like round yellow and wrinkle green, and what he get in the first filial generation are round yellow seeds only, because round and yellow are dominant over wrinkled and green. And then with the Punnett square, when he self-pollinate it to the F2 generation, he got the answer as 9:3:3:1. As you see, the 9 is for yellow, round yellow, the 3 is for round green, then another 3 is for wrinkle green, and then is 1 for wrinkle green. 3 is for wrinkle yellow, and then 1 is for wrinkle green. So this is the experiment showing the dihybrid cross, and as we see that here we can define the Mendel laws of inheritance. First law is law of dominance. Here we are showing one more diagram. So here also we can see the 9:3:1 ratio of the yellow and the green seeds here more clearly you see, and you can get the laws of Mendel. First law is the dominant law of dominance, in which one character is dominant over the other. Here round yellow is dominant over wrinkled yellow. Second is law of segregation. Law of segregation states that first filial generation are all of dominant, but second filial generation we get the recessive one also. That means the dominant and the recessive ones has not been blended with each other, and we are getting all of them. Third Mendel law is law of independent assortment, in which we see that one pair of character is independent to go anywhere in the second filial generation independent of the second pair of characters, and the ratio is 50-50. 50% of round can combine with this and 50% of yellow can combine with this to get this result. We will discuss more about it in detail later on. So this is for the dihybrid cross. Then we come to the incomplete dominance. These are all the laws of Mendel. The first law is the law of dominance, second law is law of segregation, and third law is the law of independent assortment. Then we come to the incomplete dominance. In the incomplete dominance we see that, as we said, that they do not blend with each other. But what happens? They sometimes say they are blending. That's why incomplete dominance is the phenomenon in which we are getting something different from the parents, neither white nor red. We are getting something between of these, the pink one. Okay, it's a phenomenon in which the F1 hybrid shows the characteristic intermediate of the parental genes. In this process, the phenotypic ratio of F2 generation deviates from the Mendel monohybrid ratio. Example is of Mirabilis jalapa plant, the pink and the red flowers. So one more image is there in which the flowers of red and white are crossed to get only the pink flowers in the first filial generation, and second filial generation we are getting 1:2:1 ratio, for 1 is red, 2 is pink and 1 is white. Why is this so? What is the reason for this? The reason for this is that there is a partial expression of genes for these character. There is a partial expression of genes for some of the characters. Like when the gene completely expresses itself, then it gives red one. When the gene partially expresses itself, it gives pink one. And when the gene does not express itself, it gives the white flowers. What I mean by the expression of genes is, genes, as we know, ultimately they will give rise to the proteins or the enzymes, and these enzymes are only responsible for creating any character. Like if these enzymes are fully expressed, they will give the red flower. If these enzymes are partially expressed, they will give the pink flowers. And if these enzymes are very less expressed or not expressed, they will give the white flowers. So this is the reason for the incomplete dominance. Then we come to the codominance, in which we can get the multiple allele concept also, that in some it is a phenomena in which two alleles are able to express themselves independently where the parent present together, when they are present together. These alleles are codominant alleles. Like for blood, there are 3 alleles expressing IA, IB and small I. When IA is together, it will give the blood group of A. When IA and IB dominate, it will give IAB. And when IA and small I, because small I is recessive, it will only give A, and similar to that. So here the multiple alleles are expressing themselves to give the results, and 2 alleles of these multiple alleles can give any character independent of the parent. That is codominance. As it is also multiple alleles, it can also be explained by ABO blood grouping, as I have said, and this is the main basic example of multiple allelism and the codominance. We can see one more image for codominance, like for red flowers RR, R1 R1, white flowers R2 R2. When they codominate with each other, they give R1 R2 flower, which is red and white both. So this is codominance, and this is related to multiple alleles. So this is a short introduction about genetics and evolution, a chapter in biology. We will discuss more about it in detail. I hope that you have understand a little bit about what is genetics, what are genes, what are genotypes, phenotypes, what is about Mendel and its experiments, what is the conclusion you can derive from the Mendel experiments, and what experiments he has performed. Apart from that, the incomplete dominance and the codominance and the reasons for these incomplete and the codominance. So this is just a short introduction about my explanation. So feel free to contact with me. Thank you.